There we are. Hello everyone, welcome to the next episode of my science podcast. Today I'm really delighted to welcome Dr. Corrie Painter, who is a biochemist, as the Associate Director of Operations and Scientific Outreach in the Cancer Programme of the Broad Institutes of MIT and Harvard. She also leads the Angiosarcoma Project and is the Associate Director of Count Me In, as well as being Vice President and co-founder of the Angiosarcoma Awareness Inc. Previously, she's worked at a University of Massachusetts Medical School lab, where she studied the adaptive immune systems of zebrafish. So thank you so much for joining me today.
Thanks so much for having me.
So to begin with, may you tell us about your career so far and how being diagnosed with angiosarcoma in May 2010 gave you a boost to help others?
Yes, I was a scientist when I was diagnosed with this exceedingly rare cancer. It only affects about 300 people in the United States every year, so you can imagine how rare that is. And it can pop up anywhere in your body, so there's only a handful of people who had the kind of exact same diagnosis as I did. And it made me realize, since I was already a scientist at the time, that if I didn't do something to try and start research on it, then probably nobody ever would, because that's just not the way, to try to inspire people to do research, there has to really be an unmet need. And when you only have a handful of people that have something versus other diseases and cancers that affect a much larger swath of the population, of course, people wouldn't think to really focus in on something so exceedingly rare. And so when I was diagnosed, it really lit a fire to try to figure out a way not to take resources away from other larger cancers that are equally as deserving, but to try and figure out new ways of doing cancer research generally, so that exceedingly rare cancers could actually fold into the same platforms as much larger cancers, and then everybody could win.
Definitely. So what are the sort of characteristics of angiosarcoma?
Angiosarcoma is a cancer that starts in the very inner lining of your blood vessel. So if you think of your blood vessel as a hose, there's several different layers that comprise the hose. And the very inner layer that actually comes in contact with the blood, those are called endothelial cells. And that is what is thought to be the cell of origin in angiosarcomas. So when angiosarcoma tumors grow, if you look at them under a microscope, they look like just like a tanglement of blood vessels, and they'll have blood in them and everything. The more aggressive types of these cancers lose that morphology. You can't actually see them as blood vessels anymore. They're just sheets and sheets of endothelial cells that just amass into these tumors.
Okay. So we'll talk about your earlier research quickly, and then we'll move on to your most recent research. So you've done work on the plasminogen activator inhibitor 1 as a risk factor for myocardial infarction or heart attack, the interaction with HLA-DM and the MHC2 protein, as well as other things like tumor necrosis factor A and the HLA-DO inhibiting the HLA-DM function, and the circadian variation in the plasminogen activator inhibitor 1. So what came from these studies, and why did you then choose to transition to cancer studies after this?
It's a great question. So I was a technician in a laboratory down at Vanderbilt before I went to graduate school to get my PhD. And that lab was focused on cardiovascular medicine, which is where I was able to do studies with the graduate students and postdocs on PI1, myocardial infarctions. And oddly enough, one of my primary responsibilities in that lab was to develop primary cultures of endothelial cells. It's not a common thing that people study, but it was something that was part of the lab. And so I became very well nuanced in my understanding of endothelial cells, of the circadian rhythms that they express. People have heart attacks in the morning. Well, how come? Because there's a circadian rhythm and blood pressure changes and all kinds of different factors involved with that cyclic nature of the genes that are regulating the circadian rhythm are all at play there. So that experience just really made me fall in love with science. I didn't really understand what a scientist did until I worked in a lab and I started doing my own experiments, and more so getting results from my own experiments and being able to interpret them and realize that what we're doing as scientists is we're asking questions and we're moving the frontier of our knowledge as a species forward. And that alone it gave me just like a tingling sensation. I was like, oh my goodness, this is incredible. What else is out there, right? So that inspired me to go to graduate school. And when I went to graduate school, I ended up, you get to do a bunch of rotations in the program that I was in for several months. So I did several different rotations. And the lab where I felt the most inspired was a biochemistry lab that did biochemistry in the immune system. This is where all of the work around HLA, HLA-DO, HLA-DM came into focus for me. And I was really interested at the atomic level of what was going on between these proteins of the immune system. Like, what happens if one side chain of one amino acid and one protein moves 2 angstrom? I would stay up at night like, what? I was so just absolutely obsessed with this puzzle, and I loved it. And as a result of being in this lab, there was other biochemists like me, and there were straight cell biologists and straight immunologists. And being there for several years, I got the ability to filter through a ton of information about the immune system. So when I was diagnosed with cancer, one of the first things, and I was just about to get my PhD out of that lab, one of the first things that I did was start to investigate what the immune response to that cancer was, and realize that, I came across some information from a sarcoma doctor from over 100 years ago that suggested there could be an immunological response to sarcomas. So putting my background as a scientist together with this like one little clue kind of sent me on this path to try to investigate the immune system with respect to cancer. And that's what led to the postdoctoral work I did in zebrafish melanoma. I was trying to understand the interface of how immune cells migrated to and through melanoma tumors in an effort to try to develop that as a model system for visualization.
Amazing. So on that note, your work covered the suppression mechanisms of regulatory T cells, and you used a FOXP3 ortholog as a marker to identify the CD4 enriched mature T lymphocytes with properties of regulatory T cells, as well as other things like researching the CTLA4 and CD8 levels. And this was all to help determine the role of adaptive immune cells during melanoma onset and progression in these zebrafish. So can you just tell us a little bit about this before we then move on to your most recent work?
Yeah, sure. So the goal of this research really was to develop a brand new system for visualization of the immune cancer interface. So in zebrafish, there are quite a few characteristics that enable a person to look directly into a tumor. And if you can then take the T cells and the dendritic cells and the different compartments of the immune cells and make them glow in different colors, then you can actually put the fish with the tumor at different stages of the development of that tumor underneath a microscope, and you can literally just say, what do I see? What do I see? If the FOXP3 labeled T cells are more prevalent at the boundary of the tumor and you don't see any other T cells coming in, maybe that gives you a hint about the suppressive nature of a FOXP3 T cell. If you see them, but you see other green labeled CD8 T cells infiltrating that tumor, maybe you think, well, in this model system, it's not that important at this point in development for the suppressive mechanisms of FOXP3 around the periphery of the tumor, and so on and so forth. And so this work really enabled the development of a model system for you to be able to track in real time what happens as a tumor grows with respect to the interface between the immune cells and that tumor.
Amazing. I mean, that's fascinating. So my next question is, may you tell us about the Metastatic Breast Cancer Project?
Yes. So when I was a postdoc, the studies were going really well, but it became pretty clear that that goal I had, which was to try and figure out how can you study these super rare cancers without really taking away from the common cancers? How do you get anything done? I realized I wasn't going to be able to fulfill that as a zebrafish melanoma biologist. It just wasn't going to happen. So I was looking for other avenues of generating research. I was exploring everything in the nonprofit space, everything as an advocate. I was interested in just raising money and giving it to other scientists to tether them together versus having my own lab. And I came across this initiative at the Broad Institute of MIT and Harvard, where they wanted to do the exact same thing that I had been dreaming of, but they had the ability to do this at scale. They wanted a new platform to perform cancer research that included patients in the design of that research in a way that allowed the build of something where anybody in the United States and Canada could sign up online. And they would be able to sign up online in order to help us build a clinically annotated genomic database. What does that mean, right? So it is a database that contains information from a person's clinical experience that we could abstract out of medical records. It would contain information directly from a patient about their experience with the cancer. It would contain information at the genomic level about the tumor of that patient, as well as the inheritable traits in the DNA of the normal or germline cells within that person. And what it would look like is, in the actual types of sequencing that we wanted to perform was called whole exome sequencing, which is where you look at the coding regions of all 20,000 genes, and you look at them inside of the tumor, and then you look at them inside of the non-tumorous germline DNA of the same person, and then you can tell what's different. And it's the differences between what the normal DNA of that person looks like compared to what their tumor looks like that is oftentimes causing that tumor to grow and be aggressive. Oftentimes, if you follow what's going on in the tumor over time as it grows, and as that person is being treated on different therapies, you can understand what happens at the genomic level that creates resistance to therapies. And hopefully, over the course of time, if we keep looking at this and generating this data and releasing it for other scientists to also look at and study, we could better understand all the different facets of cancer. Why does it start? Why does it continue to grow? Why does it go to other parts of the body? Why does it outgrow the ability of different therapies to keep it at bay? You know, all of these different questions don't have good answers. We don't know these things yet. And we felt that if we included patients in the process of trying to answer these questions, we can get there faster than we ever have before by just having patients kind of be the passive recipients of care. So I joined the Broad to help them conceive of and launch the very first project here, which was in metastatic breast cancer. And it was, of all the cancers, started here for a variety of different reasons. The person who's leading the charge is a medical oncologist at the Dana-Farber Cancer Institute in Boston, and his name is Dr. Nikhil Wagle, and he's a metastatic breast cancer doctor. And he's also a genomicist at the Broad Institute. And as we started thinking about what a good project would be, it became increasingly clear that metastatic breast cancer would be a great place to start, right? There's a huge unmet need. People are diagnosed with metastatic breast cancer, and the average lifespan at the time was about 3.5 years. And it's considered a terminal disease or uncurable. So there's the need to change that for everybody that's ever diagnosed with metastatic breast cancer. It's a fairly large patient population with tens of thousands of people living with metastatic breast cancer in the United States. And we just thought that we could work with this patient population, work with the advocacy groups that were representing these patients in order to get the word out, to first design it in a way that would resonate with the patient population, and then work with the patients and advocates in order to get the word out that this exists and that we're trying to do good in this world and that we want to walk with them on this path toward better treatments. We try to steer away from hyperbole. So, and this was all guided by patients themselves that said, don't tell me you're going to cure my cancer in the next 2 years. Don't do it, because people have told me that for the duration of my experience with this cancer, and I haven't seen it yet, and I don't see it on the horizon. Be real with me, And so this is some of the advice that the patients gave us as we designed this with them. And we worked on this together with just dozens of patients over the course of about 10 months. And we launched the Metastatic Breast Cancer Project in October of 2015. And to date, we've had over 6,000 women and men, because men get the disease too, join and say, count me in. I want to be part of this. And as a result, we have been able to generate and release data from many hundreds of patients that now rests in the public domain. Anybody can go and take a look at this data. You can go to the mbcproject.org website and just click on data, and you can navigate to the data. You can download the data. You can analyze the data. It's free and it's not password protected because we've taken away the readily identifiable information and just have the information there in aggregate. So the hope was that people would use this data and publish on it and make discoveries. And to date, there have been over 30 peer-reviewed papers that have done just that. And if you think about what an individual lab could do over the course of a couple of years versus what one group could do to inspire a bunch of other labs, I think that it's proving out to be a success in terms of really accelerating the pace at which this research can occur.
Amazing. That is wonderful. So you've done a similar thing for angiosarcoma, haven't you, as well?
Yeah.
And when you did the whole exome sequencing, this revealed recurrently mutated genes such as KDR, TP53, and PIK3CA. So in what way did these findings suggest a therapeutic rationale?
So it was really interesting when we launched the Metastatic Breast Cancer Project, we started thinking, well, is the success of this project contingent on this patient population, or could it be successful elsewhere? And we thought that to the very, essence of why I wanted to do this in the first place, that you could take this platform and point it elsewhere and have success as well. Whether it was common or rare, we thought, well, let's test it in the rare cancer. Let's test it in angiosarcoma. You don't get much more rare than that. And we built this we used the Metastatic Breast Cancer Project and pointed it to my patient population and said, what do you think? And so they said, it's great, change some colors, let's change some words, and let's change the cover picture, and let's launch it. Let's get the data in. So we worked with the patients. We launched that in 2017. And I think success on our end would have been 50 people, maybe 100 people over the course of 100 years signing up and providing information for this rare cancer. Well, fast forward to today, and we've had well over 500 people that have joined. It's been an incredible response if you think of how rare this cancer is and how many people have joined the project. But because it's so understudied and there's so little known about it, way back at the beginning, when we first started to generate and release data, our first data release, I believe, had 14 people that were clinically annotated in the public domain. And you could see right off the bat that there was a deviation for one small subset when you looked at different genes. And we'll get to what that one subset is in a minute, but to your question about KDR, TP53, and PIK3CA, these, the KDR and the P53 had been shown previously in other genomics investigations that included angiosarcomas. The KDR is an interesting mutation in that it is a receptor tyrosine kinase. And there was a theory that if you treat with a tyrosine kinase inhibitor, you could shut that pathway down. And so this was more validation that there are people out there that have these targetable mutations in these patient populations, and they help to refine, well, which patients might those be? And so we saw both amplification of that in some of the cutaneous skin angiosarcomas, as well as the point mutations in other angiosarcomas. And so it helped flesh that out a little bit for the field. The PIK3CA is a really interesting finding here. So PIK3CA is actually one of the most highly mutated genes in breast cancers. And in our data set, what we found was that it was almost exclusively found in breast angiosarcomas. So at the beginning of this podcast, I had mentioned that angiosarcomas can arise anywhere in your body because it's part of your blood vessel. So a subset of people get angiosarcoma that arises in their breast. I was one of them. But the majority of people do not. The majority of people actually have cutaneous angiosarcoma in the head, neck, face, and scalp regions of their body, which we'll talk about in a little bit. But the PIK3CA mutations were basically exclusively found in the angiosarcomas that arose in the breast. And so that immediately was of interest to me and Dr. Wagley, who's a breast cancer, biologist, who was like, wait a minute, what? And it made you think that maybe there could be a tissue-specific niche that helps to select for PIK3CA mutations or makes them more permissive, or maybe the immune system doesn't do as good of a job for some reason, eliminating them from that tissue. So it's intriguing, and it led to a lot of really interesting hypotheses that we need to follow up still. But that in and of itself is really interesting. Now, because my background is in biochemistry and I'm a structural biochemist by training, I was able to take a look in the three-dimensional crystal structure of PIK3CA, where those mutations mapped to. Because though there were a number of different PIK3CA mutations in those breast angiosarcomas, none of them were in the same spot. There's two what are called canonical spots, canonical mutations in breast cancer. And that means that there are two spots within the gene that the majority of those breast cancer mutations are found and known to be activating mutations, turn that gene on, turn the protein on if it contains those alterations. And so I mapped the mutations that arose in the breast angiosarcomas, and they all kind of clustered around the three-dimensional space of where the breast PIK3CA mutations occurred. And so it looked like you could make a case for why each of those individual mutations in angiosarcoma, though not canonical, may lead to an activating phenotype and drive the disease. And so that then, if there was follow-up on that, could open the door to the potential of having clinical trials for breast angiosarcoma patients that have these mutations. And we wouldn't have known that before. So there's pretty significant implications clinically for some of these findings.
Definitely. So you've mentioned angiosarcoma of the head, neck, face, and scalp. And this is associated with a high tumor mutation burden. And you found that ultraviolet damage may be a causative factor and that immune checkpoint inhibition may be beneficial. So can you expand upon this?
Yeah, so it was really interesting when those first 14 patients were clinically annotated, and we saw the very first graphs in the public domain on CBioPortal. One of the graphs that CBioPortal will provide is a tumor mutational profile or tumor mutational graph, basically showing, how many samples have different amounts of mutational burden within the tumors. Mutational burden within a tumor is literally just that. It's how many mutations are occurring in the DNA of the tumor. And there is a class of drug right now called checkpoint inhibitors that are associated with having a good response in people that have a high amount of tumor mutations within their the DNA of their tumors. There are several thoughts about this. One of the prevailing thoughts is that if you have mutations in a lot of the DNA in the tumor, the DNA turns into RNA, turns into proteins. And if you have a bunch of proteins that perpetuate what that original mutation has, the protein that you end up with does not look like you to your immune system. It's different than what your immune system was trained on when you were a baby and your T cells were going through your thymus and trying to get a sense for all of the different permutations that may pop up that it needs to watch out for, because your immune system is trained not to attack you. And in this instance, when you have an accumulation of mutations in the tumor DNA, that turns into a lot of proteins that your immune system does not recognize. A checkpoint inhibitor, what it does is it releases the brakes off of your immune system. So if you have a lot of tumors, mutations, and you've got an immune system that's literally like sniffing anything out, and without its normal brakes to like halt it from doing that, you can then get a very robust reaction to that immune checkpoint in tumors that carry those a lot of mutations. So the very first, and that's what I studied. I mean, because I studied cancer immunology as a postdoc, I was like, look at that. Oh, this is good. And so that was one of the first things we thought when we saw that. We're like, oh my goodness, what is going on here? And we noticed that there was this subset of people that had really high tumor mutation burden in that first data set. It turns out that almost all of them had angiosarcoma of the head, neck, face, and scalp region. So that led to two thoughts. The first thought was, how come? Why? And the very first follow-up hypothesis was, well, those areas are exposed to the sun, and maybe the radiation from the sun is actually inducing mutations in the DNA of the endothelial cells, which is transforming them into cancer. And it wouldn't be unlike melanoma, which is what I studied too. And so it really came down to an analysis by our computational biologist, Isha Jain, looking at the DNA in the tumor, and sun damage actually manifests within the physical bonds of the DNA itself. And you can tell by the way that the mutations, result, whether or not it was caused by ionizing radiation. And sure enough, she saw that signature in the DNA of these people who had the high tumor mutational burden and the sun-exposed skin areas. So that was a really interesting proof of concept in terms of the etiology of that particular form of angiosarcoma, that it's likely caused by sun damage. The second thought, knowing that there's this association with tumor mutational burden and this response to checkpoint inhibitors, was that not only do we have the DNA from these patients, we have their medical records. And the way that these rare cancers, sometimes these rare cancers, there is no standard of care. And so doctors will do their very best with whatever is out there in order to see if they can get a response for patients. And so we went through the medical records of the patients and found that several of the people who had head, neck, face, and scalp angiosarcoma were put on checkpoint inhibitors. And two of them were metastatic at the time, had gone through and not had any sensitivity to a variety of different therapies, and they kept progressing. And then their doctors put them on a checkpoint inhibitor, and it melted their cancer away. In two of these cases, it just eliminated the cancer. Oh, wow. And then in both cases, those men, there were two men, developed pretty significant autoimmune reactions to the checkpoint inhibitors. Because again, we've taken the brake off the immune system. And so it's now going to attack things that maybe it shouldn't. And that happened in these two instances. So those patients had to be taken off the drug, but they remain without evidence of disease many, many, many years later. And so they're effectively cured from their angiosarcoma, even though they're not on any type of therapy right now.
Amazing. Yeah. So I guess the sort of what may the future look like in terms of immune checkpoint inhibition as a therapeutic avenue for this angiosarcoma, I guess it is a possibility.
Yeah, in fact, some of the doctors in the sarcoma community, we started talking about this. I was giving talks on this. The minute we saw it, I reached out to everybody I knew. I was like, look at the data, and so it's really compelling in terms of being able to share data in advance of any type of publication, though we did end up publishing this in Nature Medicine. Before we had this published, I was out there telling anybody who would listen, look at this. And several sarcoma clinicians developed clinical trials around this and studied it clinically. And the results of those are now validating this in the cutaneous angiosarcomas. And so it has been practice changing.
Absolutely wonderful. So my final question is, what are your future research plans, and what would you like to see implemented in the way that rare cancers are approached?
I want to study all of them. I want to build out resources for every rare cancer that has not been studied to date. I think that we've shown the proof of concept that you can make a platform and you can invite people in, and that they're willing. You know, people really want to contribute to research in a way that can only be driven by them and their unique experiences and the uniqueness of the DNA that is in the tumors that they have. And now that we've put everything together, I want to see what can we do in the space of rare cancer to really drive our understanding of each of these diseases. I think that it's going to take a combination of doing kind of a very deep molecular analysis like we've done. But I think that there are so many cancers that have just not been studied at all, that just looking at the patient-reported data and the clinical information would be transformative. And so it's my great hope to be able to apply the Count Me In platform to be able to work with as many rare cancers as are willing to come on board.
That is amazing. And I really hope that it happens, and other people come on board as well, and other, it becomes a regulatory thing that everyone does, and it's used globally. That would be wonderful. And I will link next to this podcast the website that you mentioned with all the data in, so anyone listening can have a look at that. So thank you so much for doing this today. I've really enjoyed this, and it's been really eye-opening and fascinating and really interesting. So thank you so much.
Thanks again for having me. Bye, everybody.
Thank you, everyone, for listening.